Today, I had the pleasure of interviewing Mike Graffeo, CEO and co-founder of Fluidform 3D, a 3D bioprinting startup out of Carnegie Mellon University focusing on key applications using fresh technology, also known as freeform reversible embedding of suspended hydrogels. I have always been impressed with how Mike and his team navigate the challenging task of commercializing life-changing tissue engineering deep tech, and I hope this conversation can serve to guide and motivate more scientists and entrepreneurs in this space. Enjoy. All right, I think we are started. Hey, Mike.
Hey, Jenny.
I can't believe you're in my office.
It's so good to be here. Thank you
I
for having me.
know. I think we met like what, 2018?
Yeah, late 18, early 19, something like that.
Yeah, so time flies.
It's hard to believe it's been almost 6
You know,
years.
most marriages already ended by now. So, and you're still the CEO of Fluidform 3D, which is incredible because I know it's an adventure for you. And I think why don't we just start with that? I know we did a interview with Mike a couple years back, and at that time you actually had a giant a beaker, with a big heart printed in the beaker. So yeah, if people were interested, we can include a link in the notes to see what that does look like. But why don't you share with us your founding story?
Sure. Yeah. So the story actually starts a long time ago. My co-founder, Adam Feinberg at Carnegie Mellon, he and I have known each other since the 90s. We were co-op engineers together as undergraduates, and we both sort of fell in love with this idea of how do we use engineering to solve problems around the need for replacement, tissue, organs, things like that. And it led us both down a path that we've, kind of been on ever since. I spent my career commercializing new technologies in the life sciences, devices, combination drug device products, therapeutics. And Adam, went off to do his PhD, do his postdoc at Harvard, and then has been at Carnegie Mellon ever since. They at Carnegie Mellon, they spent a good deal of time in the early teens looking at the field around 3D bioprinting and asking the question, what's it going to take to make this translationally relevant? How are we going to make 3D bioprinting into something that we could actually build something that could treat a condition inside of a human? And they came to the conclusion that they needed to develop a new way of thinking about bioprinting. What they needed to do was figure out how to do bioprinting without introducing chemistries that don't exist inside the body. So, a lot of work went into developing what became fresh 3D bioprinting.
Mm-hmm.
And in 2018, they were ready to start thinking about how do we turn this into a company? How do we start doing this work at scale? Adam and I had been in touch, so I met with him, met with the postdocs and PhDs in his lab who became my co-founders. And we spent a good deal of time putting together a plan and putting together how we thought it made sense to build out a business. And we incorporated the company in 18. We started working on first getting a license from Carnegie Mellon and then raising money and getting things off the ground. And that was really how it started.
Do you still remember the first conversation you had with Adam when he first said, "Hey, Mike, I got an idea for a company"? Or did you approach him to say, "Hey, I think what you're working on is worth, a company, as a commercialization idea"?
Yeah, so there were two conversations. I remember them both pretty well.
Okay.
In 2015, I think it was, there was a publication out of their lab in Science Advances,
Mm-hmm.
and Carnegie Mellon did a lot of a PR around it. And it was sort of the first evidence that this new bioprinting technology, fresh, actually worked.
Mm-hmm.
And so I had reached out and I simply said, listen, this is the coolest thing that I've ever read about. Remember that if you're ever thinking about trying to start a company or whatnot, don't forget you've got friends in industry. Like, I'd be happy to help.
Mm-hmm.
And he wrote back, and this is very Adam about this. He wrote back and said, listen, appreciated, excited about it. The technology is going to be great, but it's not ready for prime time yet. We've got to do more work first.
Mm-hmm.
And so great, you let me know whenever makes sense. In late 17, I was thinking about that conversation. I don't know why, but it just sort of popped into my head. And I had been having the the thought process of whether I wanted to keep working in, larger companies. At that time, I was with a mid-cap publicly traded company, Insulet Corporation, where I led business development. And I was really missing my time at startups. I'd spent 6 years in startups prior, and I was starting to think about whether there was something that made sense to do. So I reached back out to Adam and I said, hey, I remember it wasn't quite ready for prime time. Would love to get an update. And he replied pretty quickly and said, how fast can you get out to Pittsburgh? I've got something I'd really like to show you.
Nice.
And so what I saw, I was out in Pittsburgh in, I think it was January of 18. And that was when, this was before the paper that came out in Science in 2019. That was when I saw for the first time the work that my co-founders have been doing, building, living, beating cardiac tissue that was recreating complex arrhythmias, and the ability to build heart valves out of collagen, and all of the work that ultimately became this, the most cited paper in all of bioprinting.
Yeah.
I get to see all of that a year and a half before the rest of the world did.
I do agree that 2018 and 2019 are the most exciting years, at least for bioprinting, because I remember your paper got published, and I remember volumetric paper got published as well, both on the cover of Science magazine within months of one another, which is quite exciting. We really thought we're going to have a beating heart that's going to be completely bioprinted back then. I mean, it's not going to happen, right? So what's going on with the heart project for now?
So there's been some really exciting developments in the cardiac space, including some work that we did at the company, building in vitro cardiac tissue. We're able to demonstrate that we can build tissue in the lab that can pick up signals in the process of drug discovery that really can't be picked up in existing assays. That's been really cool. We've got one commercial partner with that is seeing some really great results. So we really like the signal that we've gotten out of the work that we've done in cardiac, that we can build highly differentiated relevant tissue in vitro. But what's much more exciting, I think, is the work that's being done in vivo. And now we can sort of talk about it because Adam and team have finally presented on it. But there's been a consortium of researchers using our technology to develop a pulsatile heart conduit for a congenital heart disorder called single ventricle disease. And they've made enormous progress, and they've actually seen extended in vivo results that are really, really compelling. So basically think about picking up the function of the right ventricle and pumping blood out to the pulmonary system.
And this is just published.
So this is not even yet published, but just presented for the first time.
I see.
Just about a month or two ago. So
if there's any link, just share with us, and I will add to the notes and share widely.
Really happy to. Yeah, happy to.
Now, I think it will be lopsided to talk about other applications without going back to the fundamentals, which is what is fresh technology. I think I have a pretty good idea. I mean, we all have a good idea, but for people who don't know what it is, do you mind if we just kind of verbalize what it is?
Absolutely. So, the idea that came about and became the technology the company's founded upon was the notion that, if you take most of 3D bioprinting, the core problem that needed solving was biology, cells, proteins. They don't behave like plastic. When we deposit them, they don't stay where we put them.
Right.
And so most of the field has spent a lot of time working on how do we modify the inks, how do we change the chemistry, how do we very rapidly fix those things. And it's really limited the materials we can work with. And it's made it so that, it's made some very cell unfriendly environments. It's made it very challenging to do bioprinting in the way that we'd really like to. So, my co-founders spent the time to figure out, hey, what if we, instead of 3D bioprinting by depositing materials in open air, what if we immobilize those materials inside of a gel?
Right.
And by doing that, and the work that it took to figure out what kind of gel would work, and making it a yield stress material so the gel would be self-healing, but it could release at a body temperature so it was all still sort of physiologically friendly. What really became interesting was there were three core benefits of doing this sort of inside of a gel printing. Benefit one was we could immobilize those materials so they could stay in place long enough to go through the native self-assembly processes that all of biology has access to. It just takes longer than cooling a plastic down.
Yeah.
Number two is, we build those gels out of a series of, tiny little gelatin microparticles. And so we can incorporate the microparticles into the filaments that are printed. And that allows us to control a level of surface porosity in what we print that can't really be, recreated any other way. It allows us a level of resolution that's much, much smaller than the filaments that we print. And so I describe to people, think about like the dimples on the surface of a golf ball.
Yeah.
And we can sort of tune how much dimple or not we get. That patterning, that porosity that we can build allows us to control how much cellular and vascular infiltration happens when we implant these things in the body. And so that's a level of control that's really, really exciting. And then the third thing is we're able to build, inside of an environment that, outside of those microparticles is basically aqueous. So we now have a direct chemical interface with what we're building. It allows us to take advantage of not just pH-driven gelation mechanisms, but enzymatic and ionic-driven mechanisms. So we have a level of chemical control that means we can print with a much broader palette of biomaterials, and we can do it all leveraging the native self-assembly mechanism. Now, the thing that is, I think, the most powerful about this technology. I could talk about this technology all day.
That's why I'm inviting you over.
I appreciate it.
Please
Yeah.
do.
The thing that I'm most excited about and that I think is really transformative in this space is it serves as a really, really powerful vascularization plaque. So we have seen data. The first data was published in Science in 2019, where we showed, taking a small disc of collagen and implanting that subcutaneously in the mouse. When you cast it versus fresh print it with exactly the same materials otherwise, the casted material sees no vascularization in vivo whatsoever. The fresh printed material sees extensive vascularization. And that's really driven by that sort of porosity effect that we talked about, but also the native sort of chemical self-assembly that happens. So that becomes really exciting because vascularization has been the biggest challenge in all of tissue engineering writ large, not just in bioprinting, but everywhere. So that's been sort of the core unlock for what became our lead program now.
Do you remember NASA had a vascular challenge program that awarded $1 million?
I do.
Were you young enough to remember that? Because I know that's many years ago.
I was out at NASA Ames for one of the big meetings that they had. I remember it well. In fact, I do really believe that the vascularization data that we have now has far surpassed what was shown for that.
Yes.
And is a lot more clinically relevant. So it's actually, instead of sort of solving for how do we win the NASA challenge, the team at Carnegie Mellon kept their heads down and said, how do we win let's make something that can actually be implanted in the body?
Absolutely. I'm not really following the vascularization aspect closely the last couple of years, but I can tell a lot of hard work has been put in that's not totally publicized. Now, speaking of publicized fresh technologies, you and I probably on LinkedIn a lot, and you can see a lot of videos that looks just like fresh print videos. People are printing all kinds of stuff. How do you differentiate that? Because, are they using your technology or are they having their own different kind of fresh technology? How should I approach these, news or share or publications?
It's a great question. At its core, in the United States, we live in a first to file IP environment, and the team at Carnegie Mellon was the first to file. There were a few other groups that had done some very similar work.
Yeah.
And, we're fortunate to be the ones to whom the patent was issued.
Okay.
So we have the foundational IP on all of the embedded printing inside of a gel type approaches. So if you see anything that looks like 3D printing embedded inside of a gel that'll be released later out of that's effectively our IP. Now, there's a lot of groups in academia that are doing that. We love it. We think it's fantastic. It does nothing but sort of promote that this is the most widely adopted and the most powerful and useful 3D bioprinting technique in the world. And at some point, as those applications that researchers are developing elsewhere, intend to move out of the lab and into commercialization, we look forward to working with them to either facilitate us taking that in or them taking a license out to Fresh and making that available. I think there's far more applications than any one company can develop themselves here, given the power of the platform.
And you guys have the exclusive license, rights to the license.
We do.
For this. Okay, that's fantastic. Now, San Francisco and Boston are expensive cities to travel to. And you're not here to see me, obviously. You're here for the JP Morgan conference. And why don't you tell us what kind of news you brought to the conference, and what are some of the agenda on your schedule, your calendar next couple of days? You know, what are you looking forward to?
Yeah, so it starts about a year and a half ago, where there was some data that came out of the lab at Carnegie Mellon and Adam's lab that really showed some compelling reasons to believe that we could build our first therapeutic application. They had done a ton of work with the organization that at the time was known as JDRF. It's now known as Breakthrough T1D. And that had been focused on the idea that in islet cell replacement therapy, one of the big challenges is how do we get enough blood supply to the islets that we transplant?
Yeah.
And so when they saw the data out of the science paper, they said, listen, is there any way that we could work together to identify how can we help better vascularize islets using your technology? Based on the data that they saw, we spun up a therapeutic program inside of Fluid Form that's now our biggest area of focus. And so for the last year, we've been developing all of the small animal data, demonstrating that we can build, using our fresh printing technology, we can build an implant that has islet cells, that has ECM proteins, that is able to be implanted subcutaneously, that vascularizes rapidly, and that demonstrates function. So the data that we're here to promote, that we're really excited about, is our, now it's almost 4 months data, where we take a diabetes model of a mouse, we induce diabetes, we see blood sugar levels go up really substantially, really quickly. And then we do our implant, and we see those blood sugar levels come back down to normal and stay there, now out for, several months. So we've seen data that we're really excited about, and we are basically taking that now out to the next level, to move out of the small animal models into the large animal models that'll be required to go to the FDA for ultimately a human trial. So we're out here to share those data. We're actually actively raising our Series A, and so the course of the next several days, we've got meetings with investors, with strategic partners, some company presentations, and really focused on making sure that we've got the ability to go and do these large animal studies.
And you allow me to tell people how much you're raising?
Of course, yeah.
Okay, so yeah, if guys, you have $25 million, time to write to Mike. That's the best type of fundraising, just one check and done with it.
Well, fundraising is never an easy process, and it's about finding, both the capital that you need. It's also about finding the kind of partnership that you need for a long haul. These are really significant endeavors, right? To get to the stage of doing human clinicals, and then to take it out to the market and ultimately try to cure disease for a lot of patients. So what you're looking for is a like-minded group of people who believe in the mission, believe in the value, and really want to help in any way that they can.
Now, your ideal profile of investor, would this be a corporate VC, from a large company, pharma company, or it could be just any kind of traditional VC, or what is your ideal profile at the moment?
So it's a good question. The way I think about it is there's really 4 categories of investor types who have active interest in this kind of space.
Yeah.
You described there's the corporate VCs who have an interest in type 1 in general. So that's one discrete category. There's the classic biotech VCs that also makes sense. There's what I would call the sort of biology adjacent tech VCs. And then last but not least, there are, family offices and individuals where diabetes has been a big part of their life, and it's an important area to them. For us, I wouldn't say we have a preference of one over the other or an ideal. What we're really focused on is, finding the right people who share our vision that a subcutaneous treatment that is minimally invasive, is easily retrievable, and is highly engrafted, so it's viable and it's durable. That's the thing that actually opens up treatment to the millions of people with type 1 around the world.
Absolutely.
So, for us, that's the real credit. It's that like-mindedness around the mission, because I think, listen, it's a startup. Every startup hits difficult days, and that shared mission matters when you're trying to solve problems like that.
Yeah. Well, now going back to this islet cell new technology that you guys have, we actually chat a little bit before this podcast that there are a couple competitors out there potentially, and one, in fact, probably already commercializing. Would you like to elaborate on this a little bit so we have a general understanding what the space is like?
Of course, yeah. So, in this space today, there are, I would say, a few different approaches that go back some as far back as 30 years ago. So there's a commercial product that was approved by the FDA about a year ago by a company called CellTrans, the Lantidra product. And that's a human cadaver donor islets that are infused into the portal vein. And patients who get that therapy, they need to be on systemic immune suppression for the rest of their life. It's a great option for patients who have very, very poorly controlled diabetes, or maybe patients who are already on immune suppression for another reason. The majority of people with type 1 would not make the trade to say, listen, I'll give up my immunity for the ability to no longer have to take insulin shots.
Yeah.
So that as a treatment is a big step in the right direction, but it's not enough. There's other groups out there. Vertex made a very big splash in the space a few years ago, buying Sema Therapeutics, and they're definitely the most advanced of kind of the next gen of therapies. They've got a terrific portfolio of IP around the induced pluripotent stem cells and getting those to behave in an islet-like fashion. And so now they're in the clinic in what was just announced that they had done a phase 1/2, and they've now converted that into a phase 1/2/3 with another cohort. And so that's a really exciting development because unlike the Lantidra product, that product will be stem cell derived. So there's no limitation on the sourcing of it. There's no, you can only treat so many patients. It'll be a much more kind of broadly available treatment, but that still requires systemic immune suppression. Then there's a couple of categories of programs behind that, including the programs that are centered around what I'll kind of call broadly, encapsulation-based approach.
I definitely have seen that before. I think one of them is actually in the Bay Area. I forgot the name.
Yeah, so there's a few different, people have been working on encapsulation for 20, 25 years now, going back to the original islet cell transplant work where they realized, boy, if we can't protect this from the immune system, it doesn't matter what we transplant. So encapsulation, these are groups like what was formerly Sigilon, which is now owned by Eli Lilly, and the really beautiful work that's been done by the group at Aspect Biosystems in encapsulation inside of a 3D printed environment. And there's been some really exciting developments there. At its core, encapsulation always kind of competes with vascularization. So there's only so much nutrient and blood supply you can get when you're trying to encapsulate the cells. And there are, signs of promise. There's also signs of challenge, right? Sigilon had to shut down one of their major programs because of late stage fibrosis of the encapsulated cells. And, really the materials that are used for encapsulation, the long-term kind of fibrotic impact is really unclear right now. So, our belief is that encapsulation is still a challenge. It's not a solved problem, and it really won't be until you see some compelling phase 3 data.
Are these considered cell therapy in a category?
They are. It's interesting, cell therapy in our industry generally is referring to like CAR T and these sort of one time you inject the cells, they do the job, and the job is over. So we see now with the type 1 application, we're starting to see the moniker of cell replacement therapy as a little different from cell therapy. And so cell replacement therapy, the cells are designed to go in and do the job from then until forever. Or for however long that you can achieve that. So, I think we use the term cell replacement therapy. And to me, that's just sort of a bridge to going from cell therapy to tissue, right? It's ultimately, these will be tissue therapeutics. We're not quite ready to talk about that in the industry today. It's sort of one step too far for the way that most people think about it today. But I think this will be the application that's the bridge.
I mean, I think we're talking about it because, the next thing we're thinking about is the regulatory pathway, because it's such a brand new thing. Actually, a couple months ago, I saw out of Jennifer Lewis's lab, they have an implant for breast cancer that's vascularized, but it's an implant plus cells in it. I'm just wondering, like, is your islet cell implant similar in terms of the kind of combination device this is? I mean, how do you categorize it when you apply for any kind of FDA pathways? Like, where do they put you?
Yeah, so with our type 1 indication, that will definitely be a combination product.
Okay,
The Center for Biologics will be the lead on it because the cells do the primary mode of action.
I see.
But the CDRH will be involved because it's going to be cells deposited in an extracellular matrix scaffold, and so CDRH, the devices center, will definitely be involved. And for us, the CDER will also be involved. CDER, because our intent for our type 1 application is to load a local release, so kind of a depot of immunosuppressant just in the area of the implant. So we envision all three centers involved from the FDA's perspective. There are other products that have done that. It's not a first of kind. And fortunately, for us, the cells as the primary mode of action for type 1 is not unfamiliar to the agency. So we think from a regulatory strategy perspective, it makes a lot of sense to start here, because while there are definitely some open questions to answer, the road is actually somewhat clear to see how it'll play out.
Yeah. What's really funny, last Thursday we had a demo day, as you know, and a couple of investors. I heard the word Doge quite a few times. Do you think Doge is going to help?
It's an interesting question. I've been working with the FDA for 25 years. I've got 4 different FDA approvals in my career, and I probably have a different point of view on this than a lot of folks, which is I've generally seen out of the FDA that when you work with them the right way, there's a tremendous willingness to work with industry to solve real problems. So I'm not sure that, FDA is broken and it needs a lot of fixing and overhaul is the right kind of mentality here. I think in general, most folks at the FDA want to see novel therapeutics get to patients faster.
Yeah.
So I think that, are there opportunities for efficiency? Absolutely. Do I think that, the efforts that are being promoted right now through Doge will be potentially helpful? It certainly could be.
Or somewhere else, maybe for tax reasons.
Yeah, well, I mean, listen, there's inefficiency everywhere, right? It's complicated, and it's complicated for a lot of reasons that didn't just happen overnight. So are there chances to do things better? I'm sure of it.
Yeah, I mean, I've encountered people who have to deal with the EU, have to deal with Singapore government, Chinese government, all these different regulatory bodies for healthcare. And everybody pretty much agrees that FDA is probably the best entity to look up. And, I would say, okay, of all the bureaucracies, they probably actually, I mean, in my opinions, I think FDA is probably the best of all the bureaucracies that I've seen so far.
I think that's right. I think that there's, in all the different kind of mentalities, it's not just what's the paperwork required. It's kind of what's the psychology behind why it's being done that way. I've always found FDA to be very, just laser focused on, as long as we can get comfortable that the risk is managed, we want to see the benefits out there for patients.
Yeah, I think they're actually pretty innovative and try to be on top of things in terms of technology. Now, moving away from the serious technology regulatory talk, I want to go back to how you build your company, because how many people you have now in your company?
So we have 12 people today.
Okay.
We've been, a couple of different phases of the company. So one time we were a little bit larger than that. When we moved away from the in vitro models back towards the therapeutic focus, we got a little bit leaner to be just really focused on that. We've had a couple of people join the team recently, some really top tier scientists that we've been able to add to the team, which is really great.
Yeah. So when you just started with Adam, just 2 of you.
There were 3 of us at the beginning.
Okay, yeah. And then was it Andrew or?
That's right,
Andrew. Okay, yeah, Andrew. Yeah, that's right. And then how do you build the team? Because one thing I noticed about you, Mike, is you have incredible soft skills. You're a very personable person, and you always show up. I think that's like the most important thing for founders. So tell us, like, what are your challenges of building a team, and how do you make sure you have the right team behind you for what you want to do?
Yeah. Well, I think that there's a couple of things that are really important there. Number one, when a lot of people get into whether it's entrepreneurship or leadership or otherwise, you get really wrapped around the axle first on what do I do? What's the right thing to do? And the piece that I emphasize with my team, and we've got some really tremendous young early career leaders in our team that are growing and developing. I emphasize all the time to them that what you do matters, but it doesn't matter anywhere near as much as why you do it. When you care very much about the mission and when you care very much about the people that you're working with to achieve that mission, you can get away with not always doing the right thing because people know how much you care. And so they'll give you a little bit more rope, which lets you then try on and go do the things that you feel like you feel really important about with a little bit less, oh my God, did I mess it up? And I think that's really the underrated thing that we understand about the world today. When I was in my early career, people held up Steve Jobs as the picture perfect example of how to do things right. And I got, I don't know, 6, 8 years later, and Steve Jobs was held up as exactly the example of what not to do because he was this or that or the other thing.
Right.
I look back at it and I say, I think he was a terrific example of when you care that much about the product, your customers, and your people, even if you mess up a lot, and let's not say it any other way, the guy could be a jerk. Even then, the kind of loyalty that you cultivate because of how much you care is infectious. So that's really number one. Like if you're doing entrepreneurship, startup world, and you don't have that, like, I care so much about this down to my bones, you're probably not in the right spot. That's a really important piece of it.
Yeah, I'm feeling like I loved Elon Musk for the longest time until recently. I have to say he's acting a little bit out of my comfort zone, but I feel like he's the kind of person too. He answers why pretty well for the last decade or so. Except now I don't know what is going on, what's going on with him, but it's always entertaining. That said, now if you have a new member coming in to your company, how do you like mentor him? Do you advise him to read certain books to get on the same page with you guys, or get a coach, or how do you incorporate this new member to make sure this person is comfortable?
Well, I can share with you a couple of examples. The way that we think about when we hire, a talented new scientist or engineer, and she comes on board and we want to get them up to speed really quickly.
Yeah.
The first thing that we tend to do is to say, okay, we're always printing something. We've always got some level of interesting work going on in the lab, implants in the animal facility, otherwise. So we want to get them hands-on really quickly. And so it's super important to say, all right, let's get up in the lab, spend some time shoulder to shoulder with our engineers, understand why we load the syringe the way that we do, understand why we program the G code the way that we do and path things the way that we do, because this avoids this kind of trade-off and this actually sets things up better. This incorporates porosity a little bit differently. We're experimenting with this kind of shape versus that. We're experimenting with different concentrations of cells and otherwise. We want to get you in there and living and breathing the printing process. And that's in large part because we can hire tremendously talented people with backgrounds in biology and otherwise, and they can ask much better questions when they understand why the technology does what it does. So shoulder to shoulder in the lab, seeing the experiments, understanding, people have a million questions when they start.
Yeah.
They want to know why, wait, how does this do that? What's going on with this? And so all of that happens in the lab. You got to be in there. You got to have the nitrile gloves on. That's step one. Step two is spending the time with, my co-founders, with Adam, with Andrew Hudson, who've been living and breathing, how do I think about tissue engineering using fresh for now, 10, 12 years.
Right.
And just saying, all right, let's really dig in on, what this technology unlocks and how you can use it. And then, with our PhD hires, so we just brought in a really talented scientist. She's got a great background in islet cell biology.
Yeah, you said you hired a new scientist recently, right?
We did, yeah. And so one of the first things
you want to give her a shout out?
So Fatma Dogan just joined our team about a week ago. She's been a tremendous hire for us. We're really fortunate to have been able to bring her into the company. And one of the first things that we do is to say, listen, we've got a weekly science meeting. We bring all the PhDs together, and that's where we talk about, all right, data interpretation, experimental design, thinking broader picture around what are we doing next to make sure that we're asking and answering the right questions. So when we do that on a regular basis, we have the forum for that, you can really quickly get up to speed and understand, okay, these are the questions the company's wrestling with right now. What kind of input do I have on these questions?
Yeah.
And I think it's also really important to me that just structurally we create space, and we really want those questions to come out. So we try to create an environment that's going to draw those questions out. I don't like to reference Steve Jobs multiple times in a podcast, but one of my favorite leadership quotes was one of his, where he said, listen, we don't hire smart people and tell them what to do. We hire smart people so that they can tell us what to do.
I like Steve Jobs. I think he's a great guy, except I won't work for him. And also I didn't have the opportunity to work, but yeah, I love him too. So Adam is studying Carnegie Mellon at the moment. Are you guys, how do you guys get together and, continue to have the cohesiveness, even though at different lab locations?
Yeah. So, Adam's our CTO. He's been our CTO since we founded the company.
Right.
And he's a brilliant scientist who's done, some of the most advanced work in this field for a long time
absolutely.
now.
He published every single paper. I was like, I want to read that.
Absolutely.
You picked the right topic.
His lab they do phenomenal work. And it's his lab is by and large leveraging this technology in most of what they're doing. So it's also really fertile ground for early stage R&D for what the company will be doing next.
Yeah.
So if you want to know what Fluid Form's going to be doing in 3, 4, 5 years, go take a look at some of the things that are happening inside the lab at Carnegie Mellon.
Well, he published a paper focusing on AI and bioprinting a couple of years back.
Yeah.
So
yeah, one of the earliest ones in the field.
So I wonder when you guys going to use AI to put AI in your pitch deck.
Well, stay tuned. Right 2025, we've got some really exciting developments. There will be several things on that front.
Look forward to it. So. Well, you have so many things to do in life. How do you balance I'm glad. When you first wanted to start the startup, kind of high risk, I would say.
Everyone is, yeah.
How did your wife respond to it?
I am incredibly blessed to be married to a partner who has a lot of patience and understanding around when I get lit up about an idea, like letting me run with it. So we had a long talk. I told her I was missing startups and I think maybe I wanted to do it again, and she wanted to ask the questions like, help me understand what it is that you're thinking about and why, and what is it that makes you feel like that's the right thing for you. And when we had that discussion, she was really supportive of it. So I feel really blessed that she was able to say to me, look, if this is what you got to do, like, go do it, but just go do it well. And so having that kind of support and partnership has made a huge difference.
It is. That is probably one of the few choices in life that's really impactful in our lives as our life partners. Okay, well, final question for you. For 2025, okay, I personally think the source of information is extremely important for our daily life and decision makings. What are some of the recommendations you would have for people to get the best information available, either for bioprinting or biotech or your life in general? Do you have any recommendations for people's reading list, podcasts, YouTube channels and stuff? Just throw it out there.
Sure. Well, first off, if you're interested in learning about bioprinting, I would start with the Fluid Form social channels.
Okay, yes, we'll share the link.
We definitely have, we've got some really good material there. There's more coming. We're going to be able to be a little bit more public about the work that we're doing and why we're so excited about it. So follow along there, Twitter, LinkedIn. And of course, if you're looking to know what's going on in the 3D bioprinting world, even larger than just Fluid Form, you should be following along with 3D Heal.
Oh, thanks. That's very kind of you.
And then, I think just broader, so when it comes to what's going on in biotherapeutics writ large, I get most of my daily updates from the group at Stat News, which is a subsidiary publication out of the Boston Globe. The reporters there do an awesome job, and it's really easy to stay on top of what's going on in the industry. Kind of the single source there. There's other really great resources, Endpoints News and others that are terrific. But I think that, picking one or two of those and staying on top of it really helps. For me, the other thing that I think about is, what am I doing to cultivate, the sort of sense of steadiness in the world? And I've spent a lot of time at the intersection of, different philosophies and approaches around this. But I think whether you're getting some daily psychological nourishment from, somebody like Ryan Holiday and the
Yeah.
daily Stoic practices, or, Buddhism or religious traditions or otherwise, the consistent theme of, remember, there are things in this life you can control and things in this life that you can't, and you really ought to be focused on the things in life that you can control, which
Mhm.
largely exist between the ears.
Yeah.
That's really nourishing and really helpful because,
Yeah.
it's all hard. There's no easy path in anything anymore.
Do you find different books to get that piece of philosophical calmness, or do you just go to a one source, that like a set of quotes or app or something for that kind of comfort?
Yeah, so I love reading books. I read all kinds of books. If Ryan Holiday publishes something, I'll read it. He's a
He's a
must
great
read as far
writer.
as I'm concerned.
Absolutely.
I think that, there's a lot of that message woven into, if you like podcasts, woven into various of the Tim Ferriss interviews as well. Several of those are great. And I think that when depending on a religious inclination,
Yeah.
most of the world's religious traditions also teach that in one way or
Right.
another.
Exactly. Yeah, I agree. I think religion actually on fundamental stuff is similar to one another.
Absolutely.
Yeah.
Yeah. Be a good person. Try not to worry about the stuff you can't control. These are universal things.
Yeah. I actually just finished the book, How to Think Like a Roman Emperor. I don't know if you've heard of that
I've
book.
heard of it. I've not read it.
It's a newer version of a regurgitation of Marcus Aurelius' meditation.
Mhm.
And that's exactly what I said. The goal is actually not to be happy. The goal is to be a good person.
Yeah.
So I will end on that note. Thank you very much, Mike, for joining us in the studio today with our duct taped camera and overnight shipped microphones. Thank you very much.
Thanks for having me.
Yeah.
It's been great.
Come by next time.